So we were talking about the recycling of these epithelial cells and the continual makeup of new epithelial cells in the crypts of Lieberkuhn. And if we were to take a look at the brush border of the small intestines on microscopic inspection, it would look like this under normal circumstances. But in a person with gluten enteropathy, you would see a great disruption as a result of inflammatory processes inside the lumen. You can see both a shorter and less numerous villi. Now, these next several figures are some of the best figures in this entire chapter related because they summarize several of our digestive processes. What is being digested, what secretions are involved, and then what is being absorbed. So our dietary carbohydrates, many of our dietary carbohydrates are taken in as polysaccharides like starch or glycogen, mostly starch from plant material. But if you were to have fresh meat, it might have some amount of glycogen polysaccharide, which could also be broken down. These polysaccharides are broken down by amylase. Amylase, so named because some of the principal starches it breaks down are amylose and amylopectin. So we secrete amylase from both salivary glands, so we can start starch digestion in the mouth, and it continues in the duodenum where we release pancreatic amylase. So starch is broken down into the disaccharide maltose. So our diet also includes a number of disaccharides, including lactose or milk sugar, and also sucrose, which you'd find in just your general table sugar, a common sweetener. And I would like you to know what monosaccharides make up each disaccharide. So for example, with maltose, each maltose molecule is made up of 2 glucose molecules bound by an ester bond. In lactose, we have glucose and galactose bound by an ester bond. And in sucrose, we'd have glucose and fructose bound by an ester bond. And it's also important to note that we do have in our diet some amount of monosaccharides too. For example, it would be very common to find fructose in sweet-tasting vegetables and fruits. Now, the enzymes that break down these disaccharides are embedded in the microvilli of the small intestines. So the enzyme that catalyzes the breakdown of lactose is named lactase, and in individuals who have lactose intolerance, they do not have sufficient lactase to break this down, leaving lactose in the lumen for bacteria in the lumen of the GI tract to use it as a fuel source, creating some gas and gastrointestinal discomfort. Maltose, the breakdown of maltose is catalyzed by the enzyme maltase, and the breakdown of sucrose is catalyzed by the enzyme sucrase. Now, for glucose and galactose absorption, they are absorbed through our SGLTs, or sodium glucose cotransporters. No energy is used right at this transporter, but because they are co-transported with sodium, it does require that we maintain low sodium concentrations inside these cells, which requires sodium-potassium pumping on the basolateral side of these cells. This means that the most appropriate term for this type of transport process, again, is secondary active transport. Fructose is absorbed through one of our glucose transporter molecules, GLUT5, which can co-transport multiple types of sugars. And fructose then can enter in a non-energy requiring process. All of these monosaccharides can leave these epithelial cells through GLUT2 molecules on the basolateral side, where they can be absorbed into our capillaries. For protein digestion, you'll recall that we start protein digestion in the stomach through the actions of pepsin, which is secreted in as pepsinogen and is activated by hydrochloric acid and itself. And then we also have our pancreatic proteolytic enzymes, which are secreted from the pancreas into the duodenum, secreted in their inactive forms of trypsinogen, chymotrypsinogen, and procarboxypeptidase. They are then activated inside the lumen of the duodenum, initially by enteropeptidase, activating trypsinogen to trypsin, and then by trypsin for these others. Once active, they can break peptide bonds in these proteins, breaking them down into further smaller and smaller small peptides and amino acids. Our absorbable units for proteins are going to be the amino acids and some extremely small peptides. Embedded in the microvilli, we have some aminopeptidases which can break down some of these very small peptides and allow us to then absorb those amino acids. Should we absorb some of these small peptides, they're typically broken down inside the epithelial cells by intracellular peptidases. It's important to note that the proteins that we break down in the lumen of the small intestines is not just your dietary protein, but with that regular turnover of these epithelial cells, there's going to be a great deal of proteins available for digestion from those cells. And it's important to note that each of these digestive enzymes themselves is a protein, which will eventually be degraded, and we can use those amino acids as well. Most amino acid absorption is going to be through secondary or tertiary active transport, being co-transported with either sodium or with hydrogen ions in the case of some of our small peptides. And because of that, we need to maintain low sodium concentrations and low hydrogen ion concentrations inside these cells. And then the amino acids can then enter our capillaries. For fats, we'd have dietary fats often coming in small droplets with triglycerides, but in the very aqueous environment in the small intestines, you would find that those fat droplets would tend to congeal together, much like when you try and mix oil and water together. These lipids will be emulsified by bile salts, and lipase can act upon the great surface area of these and break these down into monoglycerides and free fatty acids. Along with bile salts and lecithin and cholesterol, we form these micelles, very small molecules on the order of a couple billionths of a meter, less than 5 nanometers in diameter typically. We can absorb the monoglycerides and free fatty acids into the epithelial cells, and then through the actions of the smooth endoplasmic reticulum can be converted back to triglycerides. Now, once we form these triglycerides, it's important to note that these triglycerides have important nutrients that our cells could take up and utilize, but this is going to be hydrophobic. And so in order for this to be transported in the circulation, it's going to have to be bound with some other type of molecule. And in this case, it will be associated with various lipoproteins. These lipoproteins being amphiphilic, kind of like bile salts or phospholipids, encoding these triglycerides, forming what we call chylomicrons. These chylomicrons have similar constituents to what you've learned about when you learned about high density and low density lipoproteins before. It's got some cholesterol, it's got some fats, and it's got some lipoprotein. These chylomicrons can leave these cells via exocytosis and enter the central lacteals. The central lacteal, remember, is a lymphatic vessel, and eventually it will empty by thoracic ducts, the subclavian veins, into our general systemic circulation, where other tissues can then take up fats from those chylomicrons. So a reminder of what we absorb. We absorb monosaccharides, amino acids, and maybe a small number of very small peptides, and fatty acids and monoglycerides. These started as often as polysaccharides and disaccharides, proteins, and triglycerides. One important micronutrient that's absorbed in the small intestines is iron. So a lot of dietary iron is not going to even be absorbed and will simply be lost in the feces. Absorbable iron is typically in one of two forms, as heme iron or ferrous iron. And they can be brought in via various carrier molecules into our intestinal epithelial cells. Once it enters here, any iron that we need can be exported from these cells via a protein called ferroportin. I mean, what's a good name for a molecule that serves as a port for iron? Well, ferroportin. And iron then can enter the circulation and be bound with transferrin and transported to the bone marrow, where it can be used to help make hemoglobin to pack into our red blood cells. Iron that isn't needed combined with ferritin, and that will just stay here, and eventually this cell will be sloughed off the tip of the villus, and that iron will eventually be lost also in the feces.